Okay, so I would like to start off by acknowledging that we are meeting. This meeting is being hosted on the traditional lands of the Bindal people and pay my respects to elders past, present and emerging. And if you on Zoom want to ask any questions, you can either unmute yourself at the end of the presentation, or you can put the questions into the chat box and we will read it out to Andrew. Without any further ado, I think I will give the word to David to present. Andrew.
To introduce Andrew, yeah, thank you, Inga. I'm still not really sure why it's me introducing. Although he told me this morning that he might have nominated you because I thought you might say something interesting. I'm not really sure that's a compliment, but there we have it. So I don't like long intros. I've got a very short CV for Andrew. Andrew is a legend. He is unquestionably a legend. As a coral biologist, his credentials are absolutely impeccable. He did honours with Terry in 1994, and his thesis, his PhD in 2001 was with Terry and Betty on rival ecology and dispersal. He did a postdoc, a JSPS postdoc in the Ryukyu University in Okinawa, working on larval physiology, which I guess was at Suzuko with Sakai et al. He had a future fellowship on climate change, and he's been a Coral Centre CI since the outset. So under the heading other stuff, I have written that Andrew knows everybody, which is absolutely true. You go to pretty much anywhere and, oh, Andrew Baird, yeah. He's a legendary guitar player, legendary in inverted commas. I've experienced this kind of firsthand, but at a distance. He travels a lot, has spent more time in the water than pretty much everyone else, and including 2 expeditions along the length of the GBR. And he has ex-students and collaborators pretty much everywhere, every continent but Antarctica. He's got more citations than me. And he's spent more time in Okinawa than me. I admire the sort of low-budget, thinking-intensive science that Andrew used to do, although that's kind of changing with Project Phoenix, as you will see. So his kind of low-budget science is in stark contrast to the kind of thing that we do, which costs enormous amounts of money. His current interests are systematics, biogeography, and in particular taxonomy, and that's really where he's headed. So Andrew's talk today is, when does taxonomy matter? And it's part of the Project Phoenix crusade, in which Peter Kalman and Tom Bridge are his main co-conspirators. You'll see my name at the end there, which is I'm kind of peripheral to all of this. I'm really looking forward to what Andrew has to say today, because I've been finding myself recently arguing for diplomacy in this area, and I'm not sure to what extent Andrew's compliant. Will I do my job?
So I was right, he did say lots of interesting things. Yeah, no, thank you, David, and thanks everyone for coming. The collaboration with David is quite recent, and I must admit, I do value David's opinion, and I also value his attempts to rein me in, and I hope he realised I had taken this on board today, but David can be the judge. I mean, again, I thought initially a little bit of controversy generally helps, and certainly that's the sort of thing you learn in the media workshops and things like that. But I also accept that there does come a time when you've got to try and get everyone on the same page, because and being overly critical doesn't necessarily help there. But on with so the title is, when does taxonomy matter? And we'll explore that question today. I mean, again, I think there are very few occasions that it doesn't, but there are. Again, I think whatever your research question is, you really need to be sure in your mind whether or not it does. Okay, so look, I hoped not to have to present too much taxonomy, but as I put this talk together, I realised I've got to give you some sort of introduction so you know how it developed and how it developed in the way it did. So I'll give you a brief introduction to the 250 years of taxonomy that culminates in the Veron 2000 Corals of the World framework, which is essentially where we've all been working for the last 20 years. And some of the subsequent tests of the validity. So, and then I'll present a number of case studies to show to highlight when taxonomy does matter, and then a couple where it doesn't. And we'll focus in particular on trying to work out the taxonomic identity of about 30 recent genomes that have been sequenced, coral genomes. And then I'll finish up with a brief summary of what we all think is the way forward. So the first coral species were described by Carl Linnaeus in 1758. I mean, this was the beginning of the binomial nomenclature, essentially the start of taxonomy. And on the left there, you can see the Latin in which Carl Linnaeus wrote. And you probably won't be able to see that, right? It's pretty terrible, but amongst others. Now, Linnaeus based his description on a photo that was published in another paper by a guy called Rumphius, who was working in Indonesia. It's not sure whether Carl Linnaeus ever actually had an original description. So for this species, whether that specimen, the holotype, got lost or not, we don't know. But when Carden Wallace came to write a revision of the genus Acropora in 2000, one of the reviewers pointed out that we really need a type species for this genus. And so Carden designated a neotype based on that image. And so you can see it's a pretty good match. And she also went to as close as she could get to the type location of Rumphius's drawing, which was, I think, in Banda in Indonesia. So anyway, so there's 250 years of coral taxonomy there, starting with the very first description from Linnaeus. And then over the next 230 years, you know, a number of people did significant work. And in the course of that 230 years, 2,300 nominal species were described. And it was essentially a process of just adding to the nominal species. There were a few revisions, and I'll explain what they involved down the track, but essentially just the number of nominal species building in time until the first major revisions of the order were done. And they were done here in Townsville by a handful of taxonomists. You know, over the course of 8 years, Charlie Veron and his collaborators put together 5 volumes. And in the process of putting together this work, they reduced the 2,300 nominal species out there down to about 700 species, 700 accepted species. So this is the way taxonomy works, right? And so there's 1,600 synonymies and species that they couldn't work out in the order. Now, I'll just give you a brief lesson in taxonomy. This is the lesson that I wish I'd got when I did coral biology back in the 1980s, because when we were presented the ID guides, we had no idea what the work that had gone on behind them. And so a nominal species is forever. When you name a nominal species, you generally assign a holotype, and that name never changes. It never disappears. It's there forever. However, in the course of that 230 years, people working in different parts of the world, often people will describe the same thing twice and they'll give it a different name. And so that's what synonymy is about. Someone else comes along later and they look at these 2 specimens and say, look, actually, they're the same. So they synonymize the later one with the earlier one. And here is an example of that. So on the left, we have Acropora millipora, described by Ehrenberg in 1834. He didn't record the type location, which can be a problem. Here's Acropora spathulata, described by Brooke in 1891, type location, the Solomon Islands. Now, Charlie and Carden in this case, in one of those volumes of the Scleractinia of Eastern Australia, they looked at those two holotypes, or pictures of them, and they decided they were the same thing, right? And so they synonymized Spathulata with Millipora because Millipora was the first one described, and so it takes precedence. Then in 1994, Betty and Carden, doing their work on hybridization, realized that there were actually two morphs of what they were calling Millipora out there in Orpheus Island, and they didn't breed. So, you know, they tried to cross these species and they didn't. There was a firm breeding boundary. And so on the basis of that added line of evidence, they resurrected the name Spathulata. So they resurrected Spathulata. And so you can see that, so through the course of time, the number of names in different categories changes. So in 1834, we've got the one nominal species. In 1891, when Brooke describes Spathulata, we've got two nominal species. 1984, one synonymized. So there's still two nominal species, one valid, one synonymized. In 1994, the second one resurrected. So we've got two nominal species, both valid. And so here is a diagram of the number of Acropora names through time. The red line is the number of nominal species, and you can see that continues to go up. That just goes up and up and up and up. A nominal species is forever, whereas the number of accepted species varies up. It goes up and down. And so you can see that even Dana was doing the odd bit of synonymies because the accepted species isn't increasing at quite the rate. But you can see with the work in 1984 that there was this huge drop in the number of accepted species. And again, when Wallace put out the latest revision of this of the Acropora in 2012, she synonymized a few further. So yeah, I think I've got a summary of the Acropora. There's about 400 nominal species, of which only 130 of them are accepted. So when I started here in 1986 at James Cook University, this was the text that we used. This is Corals of Australia in the Indo-Pacific. And that was, you know, that's what we used all through the 90s until 15 years later, Charlie put out Corals of the World. And so this is, 2000. And this is the work that all of you guys, any of you that work with corals will be familiar with. And, you know, again, when we did our courses, we weren't taught taxonomy, we were taught coral ID. And so this was presented to us as the gospel truth. And yeah, we had no idea of, as I mentioned earlier, that the turmoil behind it, all the synonymies, all the taxon that people just couldn't work out. But anyway, so here it is, Charlie's hypothesis of the corals of the world. He listed 18 families, he listed 112 genera, he listed 799 species. Not only did he put out that, you know, his idea of the evolutionary relationships implied in that hierarchy, family, genera, species, he also put out distributional maps for every one of those 799 species. And so here's just an example of Acanthastria baobanki, and you can see that there's its distribution restricted to the sort of central Indo-Pacific. He also put out a family tree of phylogeny. And again, this is also based on fossils, so you can see it going back to whenever, ending up at the top here with our 18 coral families. Okay, now, I think it's important to understand a number of things about all of the work that went on until about 2000. There was no molecular data. There was no quantitative morphology. They didn't rely on any other lines of evidence apart from the reproductive, including the reproductive biology, which again can be incredibly informative. There were no experimental tests of the key concepts, such as morphological plasticity, which I'll bring up again later. And to be honest, when you go through the monographs, the series of books that started in 1980, the Scleractinia of Eastern Australia, it's clear they didn't look very closely at a lot of the type material. Now, difficult, 2,600 species scattered in about 40 or 50 different institutions all over the planet. It's difficult, but again, a lot of these synonymies were based on just looking at pictures in somebody else's book. It wasn't particularly rigorous. Okay, so since 2000, with new techniques, people have been testing these ideas. Now, this is a figure from Alex Kerr's paper in 2004. Alex put together a super tree, which is a tree based on both molecular and morphological data. This tree, the first inkling that things were wrong was in 1996 when Palumbi and Romano put out a paper that showed that there were these two deep clades. So you've got the robust and complex clade, which Alex's super tree picks up here. But the other thing that Alex's tree picked up was that species from the family Caryophyllaceae is the example here, were distributed in 4 different parts of the tree, 3 different parts of the tree, right? Now, work since then, there was another paper by Fukami and others in 2004, has since shown that none of those 18 families were monophyletic. Now, in your phylogenies, you want them to be monophyletic, which means that your clades contain all of the species in that group and only the species in that group. That's a good phylogeny. And yeah, none of the 18 families were monophyletic. Now, Charlie 112, in Charlie's hypothesis of the number of genera out there, has since been tested multiple times. And this is an example of work that's been done within the genus Montastria. So Charlie recognised 12 species. Those species have since been distributed among 6 different genera in 3 families. Now, people haven't looked at every one of the 112 genera, but whenever they have, the genera are not monophyletic. So not one of those 112 genera is likely to have been correct. Yeah, anyway, so that is right. And this brings you to some of the work that we've been doing. So now we've been testing the genera Acropora. So, so Charlie recognised 132 species. There's 402 nominal species. So there's 280 species that are not even listed in corals of the world. And so this is the work that I have been doing with Pete and Tom. The key to this work really was a couple of things. One, going back and collecting all of the type material. And that's what I did, visiting museums all over the planet over the last 4 or 5 years, getting good images of all the type material. And the second most important aspect of this work was new molecular techniques, which actually do seem to have the power to resolve species level differences. Until very recently, that has not been possible in groups like the Acropora. Some markers have worked in other groups, but the problematic speciose groups like the Acropora, Montipora, Parietes, until the UCEs, the targeted capture of UCEs and exons, until this method came along, no one was getting good species level resolution. So what we found is that with 890 specimens, is that there's a big 6-clade structure to the Acropora. We've since added another 500 specimens, and that clade structure seems robust. In our 890 specimens, we've got 272 primary species hypotheses. That's sort of where we start. And 50 of those correspond to accepted species, which means that they look like the type material. We've collected them from the type location and they look like the type material. So we're pretty confident they're good species. A lot of the species that were previously synonymized also look good. Again, testing them the same way. We collect from the type location, we compare it to the type material, and the genes tell us that they're different from other things. There's 122 species that we can't match with any type, that are genetically distinct and we can't match to any type material. There's 122 that might be synonymized species or might be accepted species, but until we get the decent type, the topper type material, until we collect from the type locations, they're unresolved. And then there's 72 that look nothing like anything out there. So there's at least 72 of which we are strongly convinced that they're undescribed species. So, we think that rather than the 112 species that were recognised in the last revision, there's probably closer to 400 or 500 Acroporas out there. So when you go into the field and you see a colony and you go to the book It's no surprise it's not there. There's only 1 in 5 species that are actually in the ID books. So look, just to give you an example, so I've been working at Pelorus Island forever, and you know, we just assumed that there was just one dominant table species, you know, 1 or 2. And what our work is showing is that there's actually 5 different species that a Veron and Wallace trained person would call hyacinthus at Pelorus. So just southeast Pelorus. And there they are, you know, with our provisional names. They're genetically distinct, and if you look closely, they are usually morphologically distinct as well. One of them, the one in the middle there, Pectinata, corresponds to a type species that was named on the GBR by Brooke. Hyacinthus is here. We've got a topotype for Hyacinthus from Fiji, and this one is genetically close and it looks the same. And then the other ones are almost certainly undescribed. We can't find any material. They're genetically distinct. We can't find any type material that they look closely. And if you look at Hyacinthus on a global scale, it's probably about 20 different species. So again, another test of the range sizes in Charlie. So, you know, not only did he describe all these species, but he put out their range sizes. And so what I've done here is other data that Charlie put out called Coral Geographic. I've just looked at the 20 most widespread species, and sorry, this is really small, but again, not all of these distributions have been tested, but every one that has shown that these widespread species are actually species complexes. So, for example, Acropora damicornis, throughout that range, it's at least 4 species. Acropora hyacinthus, as I was saying, throughout its range, is probably closer to 20 different species throughout the range that Charlie hypothesized. So again, you know, the biogeographical data is also, it hasn't stood the test of time. Those hypotheses have not stood the test of time. Now, this is a list of about 20 papers, all published by the centre, some of them very high profile, 5 of them in Science and Nature, which used the Veron biogeographic data set or data that we collected within the Veron framework. Now, so yeah, there's a lot of science out there that needs to be revisited. And yeah, so exactly what are the implications of working within a flawed taxonomic framework? So I've already touched on the range sizes one. So this is work that Terry published in 2002 in Ecology Letters, and they compared the frequency distribution of range sizes of fish and corals and suggested that there was something special about corals. As you can see, the top graph, it's right skewed. You know, it seems that most corals have these really widespread ranges compared to fish, which are more evenly distributed. But as I showed you, all of those widespread, most of those widespread species are species complexes. So again, if we revisited this data, it'll be years before we could do it properly. I suspect, you know, there's nothing really that different between fish and corals in terms of their range size distributions. This is another paper, a science paper that we published in 2003. And in this figure, we made the point that there's 24 species. That's what those contours are. There are 24 species that occur both in the Red Sea, in the Gulf, that little 36-degree dot up there on the left, and does that come in there? No, and Lord Howe Island, right? And so the Gulf average annual temperatures 36 degrees, Lord Howe Island 24 degrees. 24 species have ranges that encompass that range in temperature, and so we use this data to argue that, you know, lots of species could handle climate change. In fact, our Acropora data shows that there's not a single species in common between Lord Howe Island and the Gulf, and I'm reasonably confident that that will hold for all of the other taxa that occur in the Gulf. So yeah, so yes, this sort of assumption certainly needs to be revisited, and indeed, about 30% of the species on Lord Howe Island don't even occur on the Barrier Reef. So that's how badly we misunderstood the biogeography. This is another interesting experiment looking at thermal tolerance of Orbicella. fabulata. And so this one group did an experiment. They measured the thermal tolerance. And this is in terms of loss of densities of symbionts after subjecting it to stress. They were interested in estimating heritability, and heritability estimates are determined by variation in this trait. They got they got a lot of variation in the trait, and so they made the projection that there was sufficient variation in this trait to enable this coral to adapt to 2 degrees of climate change. Then this other group came along, looked at all their specimens, and realised that they're actually dealing with 3 different species. They've got them 3 different clades, potentially 3 different species. And if you reanalyse the data, there's actually very little variation in thermal tolerance within each of those morphs. And so therefore, it's very unlikely that any of these guys could adapt to climate change. So here is an example of where taxonomy is absolutely crucial. The answer you get is exactly the opposite. Connectivity. Again, this is some work Terry did, 2004, I think it was published in Evolution, looking at connectivity, 5 different species between Lord Howe Island and along the Great Barrier Reef. I don't know if you can see the species names there: P. dam, S. hystrix, S. pistillata, Cuniata vallida. They're all now known to be species complexes within this range. In fact, I'm pretty sure neither Cuniata nor vallida even occur on Lord Howe. Now, I think these data were done with allozymes. So, just proteins, and everything's got proteins. And so, you whack this in. Well, you whack it in, you get an answer. But the interesting thing is that when you talk to population ecologists, you would think if they were different species, you'd end up with just really big differences, right? No connectivity, but that's not the way it works. And in fact, it's very difficult to interpret these. If you're not working with species, it's almost impossible to interpret this data. So I'm told, and there was a paper published just the other day that Tom sent me on tenuis in the Western Indian Ocean. Tenuis is in Fiji, it doesn't occur in the Western Indian Ocean. It's at least 2 species that they were working with. Who knows what the data means? Conservation and management, and in particular extinction red list. The IUCN red list lists species in terms of their vulnerability to extinction. It's pretty meaningless if you can't identify species. I mean, the way the red list is meant to work is looking at changes in abundance through time, and if species are declining by 30%, yeah, put them on the red list. You can't, if you don't know where the species boundaries lie, you can't get good estimates of abundance. And so realistically, we need alternative ways to protect our reefs than the red list. And there are many out there. So ecosystem approaches. Now, to the genomes. Okay, so the graph on the left there is all of the 30 odd genomes that are out there. So what Pete did was he uses our targeted bait set to challenge these genomes and grab down all the loci that we've got. And so he ends up with, I'm not sure when the more inclusive the tree is, the more different taxa it has, the fewer loci you get. And then you throw them into a phylogeny, and so that's what you get on the left. You get this include the ones up the top of the Acropora, Montipora, and then down. And so it shows that the UCE approach is very useful. We can harvest information from these genomes, and then we can throw it compare it to our specimens. So the one on the left just compares all the genomes, and the one on the right actually introduces our specimens. And we've only been working meaningfully, we've only got enough samples to make meaningful conclusions with the Acropora. So the second graph just includes the Acropora. And the other thing to notice about the graph on the left is how little divergence there is amongst all the Acropora, right? So that's one of the reasons we're having problems. One of the reasons previous molecular approaches had been unable to find differences among the Acropora. They've all, or many of them, have evolved quite recently. So I'm now going to go through these genomes just in a PDF because the PowerPoint didn't work. I'll just go through them one by one and sort of explain how we go about trying to identify these different genomes. Now, The most important tool we use is the topotypes, right? So all the topotypes are in bold. Now, a topotype is a type that's collected in the type location and looks as similar to the type material, to the holotype, as possible. And so we went to Fiji, we collected something that looks exactly like hyacinthus, and we got its, its genes. And so you can see that the hyacinthus, the refuge, all the genomes are in blue. Not COVID. It's a cold. And again, the other thing about this tree is that we've generally only got one specimen per species, and we don't have all our species. So even though the two things can occur right next to each other, they're almost certainly different. Now, this refuge genome, certainly it has affinities with hyacinthus, but there's a lot of stuff in between in our full tree. And if you look at, so we've also sampled from the Great Barrier Reef, which is where that hyacinthus genome comes from. And we do think it's close to this specimen 733864, which I can show you an image of, but it doesn't look a lot like hyacinthus. So anyway, we're reasonably confident that that is not hyacinthus. There was a genome published quite recently by Palumbi's group. And again, you can see that it does not cluster with our topotype of hyacinthus. It's closer to another species that we've found in Palau and also on the Great Barrier Reef that we're calling hyacinthus neat. So it's almost certainly a species with some affinities to hyacinthus, but it's not hyacinthus. The oyster genome is, so this is from Okinawa in Japan. So I've also sampled in Okinawa, so 18 Oki 22. And yes, so it clusters with the specimen that I picked up in Okinawa. And again, it's not hyacinthus. So 3 hyacinthus genomes, none of which are hyacinthus. And here we have Cytherea from Oyst. And here is our topotype of Cytherea from Tahiti. And you can see that it's actually in a different clade. The Cytherea from Oyst is actually closer to hyacinthus than it is to the real Cytherea. And you can go through the tree and do that throughout. So, and there are areas where we are still not sure because we haven't sampled exhaustively. So, for example, Muricata, we really can't tell it until we get a topotype from Muricata from Indonesia. We won't know what the genes of Muricata look like. At the moment, it looks like Muricata is closer to what we have as grandis it's close to what we are calling grandis on the GBR. Yeah, we really don't know. And yes, and then here's the couple of the Millipora genomes, one of them from the Miller lab. The problem with Millipora is that it doesn't have a type location. So we'll never really know what the true Millipora looks like. And the holotype, as you saw there, it's not in particularly good nick. And so it's difficult. But, there's a couple of different clusters within, this group. We think the Millipora from Singapore is probably closest. We think it's more likely that Ehrenberg had a specimen from somewhere in Indonesia than anywhere else. And you can see that the one from Indonesia is different to the one here on the Barrier Reef. Now, there's one species on the Barrier Reef that we can be relatively certain of, which is Squamosa, which was from here, described from here. But, again, this becomes a bit of a game. What do we call it without access to adequate type material? Do we just obliterate Millipora? That is an option. We call it a nomen dubium and the name disappears. And we call it something else like Milleri.
Thanks very much.
That's right.
I think it should be, the type location should be back then.
Okay, yeah, and that's the other thing we could do is designate a neotype, and that's so we can make that the Miller genome the neotype, or we can collect a new specimen and describe it as the neotype because I think you neglected to keep a voucher. And anyway, so look, you can go through, and I think I've got a summary here. I mean, it's extraordinary. It really is illuminating to have the molecules and the topotypes, the framework to hang all of this stuff on it. It's absolutely extraordinary. Every day I look at it and go, wow, I never would have guessed that. the taxonomy is such a mess. Okay, now for some of these other genomes, we don't have enough molecular data. Obviously, we've got the genome, but we haven't been sampling intensively enough. So for these guys, we can just do traditional taxonomy and just compare them to the type material. And so this is Acropora favus that was collected by Forskull in 1775. It's from the Red Sea, and that's a photograph I took in the museum in Copenhagen. This is the voucher that Greg Torter collected for the Favia favus refuge. So this is the specimen that was sequenced to produce the Favia favus genome. And, you know, again, they're close, but not very close. And so I think, again, just the fact that one's in the Red Sea and one's on the GBR means the probability that they're the same species is pretty low. We've got, from the Acropora we've looked at, we've got 200 Red Sea specimens that we've in our tree. Not one of those clades also occurs on the Great Barrier Reef. There's almost certainly zero species overlap in the Acropora species in the Red Sea versus the Barrier Reef. That may well be different in other groups. But if it holds, then yeah, you know, the fact that a species has a type location in the Red Sea is enough to tell you that it's not here on the GBR. And this is the holotype of Parietes lutea, and truly is little. So you can imagine how little that thing is. And and some of the type material is just not. I don't know who decided Parietes lutea formed these, you know, 5 meter diameter bommies. They probably did it on the basis of what the correlates look like. Fair enough, but realistically, this sort of type material should never have been taken seriously. You know, it just should have been, forget it. It should have been a nomen dubium, and let's move on. And that happens a lot, unfortunately. And again, lots of stuff's been lost. And anyway, but so here's the summary of all of those genomes. You know, out of the 30, probably 3 or 4 have been correctly identified. Now, does it matter? In some situations, it doesn't. And so when these guys were collecting the genomes, and all they wanted was a representative of a coral, right? And so if you're comparing a coral to a worm, to see how the differences in their genomes, doesn't matter. Same if you're comparing an Acropora to a Montypora, doesn't matter. So there's lots of situations where getting it right doesn't matter. And there's also situations where the name is irrelevant too. I mean, for most of the work we're doing, it doesn't really matter what you call, or for much of the work we do, certainly not biogeography, but for organism biology, it doesn't really matter what you call it, as long as you're working within species. It doesn't matter if it's PDAM or P. verrucosa or whatever, just as long as you're sure you've got the same thing. So there are lots of situations where taxonomy doesn't matter. But again, I think, I think we can get it right. And so why not try? And so in the abstract, I mentioned, trying to understand how taxonomists, through over that 250 years, how they got it so badly wrong. I mean, and in particular, the revisions that happened in the 1980s. Now, these were the first guys in the 1980s, was really the first people to get in the water and actually see their specimens in flesh. Until then, most of the British taxonomists, I don't think ever got wet. The samples went back to the British Museum, and these guys never saw anything other than the specimens. Some of them actually travelled, like James Darwin spent time at sea. But much of the taxonomy was done without anyone ever having looked at these animals in the wild. And so this is, again, one of, in the wrong order, this is the sort of guiding principle behind all the synonymies that went on back in the 80s. They got in the water, they saw these series of forms that seemed to correspond to different environments. They interpreted that as phenotypic plasticity. So actually, I haven't got it around the wrong way. This is how it started. So when the revisions started, this series would have been interpreted as 1, 2, 3, 4 different species. Veron and Pichon looked at these guys and said, no, this is just one continuous series. And so they synonymized all of those. They synonymized those 3 other species with PDAM. And then in the 2000s, Sebastian Schmidt-Roche came along and looked at the molecules. And the current understanding of this series is that it's probably 3 species. My guess is it's probably more. Sebastian was working with single marker approaches. My guess is when we look at these with techniques that have more resolution, we're probably going to find more species of Acropora. There's something like 90 species in the genus Acropora, 90 nominal species, and only 6 or 8 that are recognised. Yeah, and so I think this was the guiding principle that led them astray. And there was also this concept that corals had these really wide geographic ranges. And again, I can't yet work out where that came from, but it's an idea that has persisted. And again, our data is showing that it's almost certainly in most situations, certainly in the Acropora, incredibly wrong. Most corals have highly restricted distributions. Okay. Project Phoenix, the way forward, reinventing a new taxonomy. It's a disaster. I don't think there's any way to look at coral taxonomy is a mess. I mean, there's been a lot of really good work that's been done. And Danwei Huang, Francesca Benzoni, they're all doing spectacularly good work and knocking groups off one by one, genera off one by one. And they've made brilliant progress over the last 20 years. But I still think it lacks a vision. And that's what we're hoping to provide with Project Phoenix. It has been stymied by the fact that there isn't adequate access to type material. So that's one of the things we're trying to do, is get all the type material, get good images of all the types, original descriptions, translate them, make them all available. And that's one thing, that's one area I think people could have done a lot better, is to make all the type material more accessible. Good molecular markers, I think we've got them, but again, I think it's also important to note that even the UCEs will have their limits. We're finding groups that they're just not working with, but there are other non-tree approaches to establishing species boundaries. So Jean-Francois Flott's been doing brilliant work with haplowebs, and you just need a lot more samples to distinguish amongst these species. So the UCEs are not necessarily going to get us species resolution in all these cases. There could be quite a bit more work that's necessary to take it to species level. Topotypes is the key. Again, without a new molecular archetype, you're never going to solve some of these questions. The bad thing is that the taxonomic code doesn't allow for you to designate a molecular archetype, and so there are barriers to progress that are imposed by this outdated code, and so that's something again that we need to address. And yeah, an integrated and quantitative approach that combines all different lines of evidence. A lot of the family relationships hypothesized by Veron were clearly incorrect just based on the reproductive biology. There were so many other things that these guys could have looked at. And again, I would say the other thing, which I don't put here because some of our collaborators don't get it, is a healthy disrespect for authority. You need to examine, you just don't take what you read in the book seriously. You've got everything is a hypothesis that's out there to be tested. And, particularly, some of these hypotheses that go back a couple of hundred years, it's proving almost impossible to get even our closest collaborators to cast aside all of these revisions. Everyone insists on working in the accepted taxonomy. So they'll go to do a revision of the genus Leptastria, and they'll go to the 5 species that are there in Veron, and I go, well, there's 20 species that were described. That's where you need to start. And of course, yeah, we're right. But yeah, so we need to get it out of our head. We need to throw aside the accepted taxonomy and revisit and test all of the nominal species. That's only beginning because there's so many species out there that are also undescribed. So it's a massive task. And yeah, we need more coral taxonomists. So thank you.